This video is audio content only. It does not contain any video footage or images. If you are expecting visual content, please be aware. This is intentionally provided in an audio-only format. We recommend listening to the audio rather than watching the screen. Thank you for your understanding. Welcome to today's exploration of one of prehistory's most distinctive creatures. Picture a beast the size of a modern rhinoceros, but bearing massive twin horns that crown its snout like ancient monuments. This is Arsinoitherium, a fascinating mammal that roamed the swamps and forests of Africa over 30 million years ago. Despite its rhinoceros-like appearance, this remarkable animal was actually more closely related to elephants. Join us as we journey back to the Eocene epoch to discover how Arsinoitherium lived, what it ate, and why it vanished from our planet's story. Chapter 1. The Elephant's Cousin in Disguise. Picture a massive beast striding through the lush forests of ancient Egypt. Standing nearly 6 feet tall at the shoulder and stretching 10 feet from nose to tail, this creature weighs 2.5 tons, roughly the same mass as a modern white rhinoceros. Its body is thick and powerful, supported by sturdy legs. Most striking of all are the twin horns projecting forward from above its nose, each one reaching impressive lengths. At first glance, any observer would confidently declare this animal a rhinoceros, perhaps an ancient cousin of the species we know today. But that observer would be completely wrong. This is Arsinoitherium, and despite its unmistakable rhinoceros-like appearance, it shares no close evolutionary relationship with rhinos whatsoever. In fact, its nearest living relatives are elephants, manatees, and the small rock-dwelling mammals called hyraxes. This extraordinary case of mistaken identity has fascinated paleontologists for over a century, revealing one of evolution's most convincing illusions and teaching us valuable lessons about how similar environments can sculpt unrelated animals into nearly identical forms. The deception begins with size and stature. When the first complete skeleton of Arsinoitherium zittili was discovered during the early 1900s and subsequently displayed at the Natural History Museum in London, visitors immediately drew comparisons to rhinoceroses. Workers at the museum initially struggled to classify the fossils, with some suggesting relationships to primitive ungulates like condylarths before the connections to elephants became clear. The animal stood approximately 1.75 meters at the shoulders, measured 3 meters in length, and possessed that characteristic stocky, barrel-shaped body we associate with large herbivorous mammals built for power rather than speed. The massive skull, robust skeleton, and heavy limbs all reinforced this rhinoceros impression. The horns complete the rhinoceros illusion. Arsinoitherium bore not just one pair, but 2 pairs of horns on its head. The posterior pair, positioned directly above the eyes, remained relatively small and knob-like throughout the animal's life, easily overlooked against the drama of what grew in front of them. The anterior pair, however, commanded immediate attention. These enormous horns rose from the nasal bones, projecting forward and slightly outward in a configuration that seemed purpose-built for combat or defense. The horns reached substantial lengths, and the gap between their pointed tips measured 40 centimeters across. When you consider that the entire skull itself measured 70 centimeters long, these horns dominated the animal's entire facial profile. For anyone familiar with the charging rhinoceros of African savannas or the extinct woolly rhinoceros of Ice Age Europe, Arsinoitherium's weaponry would seem entirely at home. Yet beneath this convincing exterior lay a completely different animal. The first clue to Arsinoitherium's true identity emerged when paleontologists examined its feet. Modern rhinoceroses, like horses and tapirs, belong to an order called Perissodactyla, meaning odd-toed ungulates. They walk on 3 toes per foot, and their ankle bones stack in an interlocking pattern that provides stability but limits flexibility. Arsinoitherium, by contrast, possessed 5 toes on each foot, the full complement characteristic of more primitive mammals. Moreover, the bones of its wrists and ankles aligned in a single row rather than overlapping, and the animal placed its entire foot sole flat on the ground when walking, a posture called plantigrade locomotion. These feet resembled those of elephants far more than those of any rhinoceros. The leg and hip structure told a similar story. While rhinoceros limbs show adaptations for speed and agility, with the ability to pivot and gallop when threatened, Arsinoitherium's limbs were built for strength and stability. The pelvis and hip joints were configured like those of proboscideans. These anatomical details painted a picture of an animal built for measured, deliberate movement through its environment, rather than the explosive bursts of speed a rhinoceros can achieve. Even the famous horns, upon closer inspection, proved fundamentally different from rhinoceros horns. The horns showed surface features that suggested they were covered by some kind of protective sheath in life, with traces indicating substantial living tissue attached to the bone.
The teeth revealed further divergence from rhinoceros anatomy. Where rhinos have lost many of their front teeth over evolutionary time and show the distinctive ridged pattern characteristic of perissodactyls, Arsinoitherium retained a more complete primitive mammalian dentition with incisors, canines, premolars, and molars all present in the ancestral pattern. So if Arsinoitherium wasn't a rhinoceros, what was it? The answer lies in a remarkable radiation of mammals that occurred on the African continent after it separated from South America during the Mesozoic era. When Africa became an island continent, isolated from the other major landmasses, a group of mammals collectively called Afrotheria evolved in this isolated environment. This superorder includes some of the most distinctive mammals on Earth today: elephants and their extinct relatives, the mastodons and mammoths, the entirely aquatic dugongs and manatees known as sirenians, the small but ancient hyraxes that look like guinea pigs but are actually closer to elephants, the strange aardvark, elephant shrews, tenrecs, and golden moles.
Within Afrotheria, Arsinoitherium belonged to a group called paniongulates, meaning almost ungulates. This clade includes elephants, sirenians, and hyraxes, united by subtle similarities in their ankle bones, teeth, and reproductive anatomy. More specifically, Arsinoitherium was classified in its own order, Embrithopoda, meaning heavy-footed in ancient Greek. For decades after its discovery in 1902, Arsinoitherium stood alone as the only well-known member of this order. Fragmentary fossils from Romania, Turkey, and Mongolia hinted at relatives, but only Arsinoitherium offered complete enough remains to understand what these animals were really like. This isolation in the fossil record meant that for much of the 20th century, paleontologists struggled to place embrithopods confidently within the mammalian family tree. Modern molecular studies have confirmed what the anatomy suggested. Genetic analysis of living Afrotherians shows that the split between the ancestors of elephants and the ancestors of sirenians occurred during the Paleocene, roughly 60 million years ago. Embrithopods appear to have branched off from this same lineage around the same time, making them a very ancient offshoot of the Tethythere family tree. The name Tethytheres refers to their association with the ancient Tethys Sea, the tropical ocean that once separated the northern continents from Africa, India, and other southern landmasses. Both elephants and sirenians show adaptations to semi-aquatic or aquatic lifestyles, and Arsinoitherium may have shared this affinity for water.
The evolutionary gulf between Arsinoitherium and true rhinoceroses is vast. Rhinos evolved from perissodactyls on the northern continents of Laurasia, sharing ancestry with horses and tapirs, while Arsinoitherium belonged to the African Embrithopoda, related to elephants and sea cows. The perissodactyl lineage that produced rhinoceroses traces back to ancestors that had nothing to do with Africa's island evolution, developing instead across Eurasia and North America in parallel to but completely separate from the embrithopods. The fact that Arsinoitherium and rhinoceroses ended up looking so similar despite evolving from different ancestral stocks on different continents represents a textbook example of convergent evolution.
Convergent evolution occurs when unrelated organisms independently evolve similar features in response to similar environmental pressures or lifestyles. In the case of Arsinoitherium and rhinos, both were large herbivorous mammals living in warm climates with abundant vegetation. Both faced similar challenges: how to process large quantities of plant material, how to defend against predators, and how to compete with members of their own species for mates and territory. Natural selection favored similar solutions to these problems in both lineages: massive bodies that deterred predators through size alone, powerful jaws for processing vegetation, and impressive cranial weaponry for intraspecific competition. But the convergence only went skin deep. While the superficial appearance remained strikingly similar, the underlying anatomy betrayed completely different evolutionary pathways. The five-toed feet reflected Arsinoitherium's closer kinship with proboscideans. The skull architecture, despite bearing large horns, showed features linking it to the broader Afrotherian radiation. Even the way the animal grew and developed followed patterns more similar to elephants than to rhinos. The discovery of Arsinoitherium fossils at the turn of the 20th century came at a pivotal moment in paleontology. Scientists were just beginning to understand how continents had moved over geological time, and the concept of island continents harboring unique evolutionary lineages was still developing. The paleontologist Hugh John Llewellyn Beadnell, who first described Arsinoitherium in 1902, named it after Queen Arsinoe, the ancient Egyptian ruler. The species name, Zitelli, honored Carl Alfred Ritter von Zittel, a pioneering German paleontologist who had worked extensively on Egyptian fossils. Early researchers, confronted with this rhinoceros-like giant, struggled to place it within mammalian classification. Only gradually, as more specimens accumulated and comparative anatomy became more sophisticated, did the Afrotherian connections become clear. The breakthrough came when researchers began comparing obscure anatomical details: the structure of the inner ear, the arrangement of blood vessels in the skull, the subtle features of ankle bones and tooth enamel. These seemingly minor characteristics, invisible in life but preserved in fossils, ultimately revealed Arsinoitherium's true evolutionary position. The existence of Arsinoitherium challenges our intuitive understanding of how evolution works. We tend to assume that animals that look alike must be closely related, that form always follows phylogeny. But Arsinoitherium demonstrates that evolution is far more creative and opportunistic than simple family resemblance would suggest. Given the right conditions and enough time, natural selection can sculpt wildly different starting materials into remarkably similar end products. A rhinoceros-shaped animal evolved in Africa from elephant relatives at the same time that actual rhinoceroses roamed Eurasia and North America, and for millions of years both flourished in their respective domains, unaware that on other continents evolution had crafted nearly identical solutions to similar challenges. This evolutionary deception also carries a deeper message about the nature of adaptation. The rhinoceros body plan, big and sturdy with impressive horns, is not unique to any particular mammalian lineage. Instead, it represents a successful general strategy for a large herbivore in warm, vegetated environments. Natural selection discovered this strategy multiple times: in true rhinos, in Arsinoitherium, in extinct South American mammals called toxodonts, and in various other lineages scattered across the mammalian family tree. The repeated emergence of similar forms tells us that there may be only so many ways to build a large plant-eating mammal, and evolution tends to rediscover the same successful designs over and over again. Understanding that Arsinoitherium was not a rhinoceros, but rather a highly modified relative of elephants, fundamentally changes how we interpret everything else about the animal. Its behavior, ecology, physiology, and ultimate extinction must all be understood in the context of its Afrotherian heritage, rather than through comparison to perissodactyls. As we will explore in subsequent chapters, this evolutionary identity profoundly influenced the animal's biology, from the structure and function of its remarkable horns to its preferred habitats and the environmental changes that eventually drove it to extinction. The rhinoceros resemblance was merely nature's elaborate disguise, beneath which lay an entirely different creature shaped by Africa's unique evolutionary history.
Chapter 2. Hollow Cores and Keratin Mysteries. When paleontologists examine Arsinoitherium skulls under museum lighting, one question dominates all others about these spectacular structures. What precisely were they made of? Not just the bone we can see preserved in fossils, but the complete living horn that projected from the animal's face millions of years ago. This seemingly straightforward anatomical question has sparked scientific debate for more than a century, and its resolution matters for understanding everything from how these animals competed to why they eventually went extinct. The bony horn cores themselves present an immediate paradox. Despite their impressive size, reaching 60 centimeters along the outer curve in typical adults and potentially extending to 1.5 meters in the largest specimens, these structures were remarkably delicate constructions. Slice through a fossilized horn core and you find walls of bone measuring just 5 to 10 millimeters thick, roughly the thickness of 2 or 3 stacked credit cards. Inside this thin shell lay not solid bone, but a complex network of hollow chambers separated by thin struts and plates called trabeculae. This internal architecture resembles honeycomb, a design engineers recognize as efficient for creating lightweight structures that resist bending and compression while minimizing mass. The smaller posterior horns above the eyes showed similar construction, though at a more modest scale. This lightweight engineering makes perfect biomechanical sense for carrying substantial structures at the end of the skull without overburdening neck muscles and vertebrae. But it creates an obvious vulnerability. Thin-walled hollow structures can be surprisingly fragile when force is applied from unexpected angles. Apply pressure in the wrong direction, and such structures risk cracking or shattering rather than bending or deflecting. How could Arsinoitherium have used these horns for anything vigorous without catastrophic damage? The answer appears written on the surface of the bony cores themselves, but reading this message requires careful observation. Well-preserved specimens reveal surfaces that are anything but smooth. Deep grooves run along the length of each horn, some following its curve, others spiraling in gentle helices. Small holes called foramina pepper the surface, varying from pinpricks to openings several millimeters across. Some stand alone, others connect to form branching networks that anastomose like river tributaries. At the base, where horn meets facial bone, particularly well-preserved specimens show a sharp lip and notably deep rugosity marking the transition zone. These features are not random damage, but epidermal correlates, traces left by blood vessels and soft tissues that once covered the living bone. During life, arteries and veins ran through these grooves and channels, carrying blood to nourish whatever enveloped the horn. The pattern and density reveals something crucial. Heavy vascularization indicates substantial living tissue attached to the bone, tissue that needed robust blood supply to remain healthy and functional. But what kind of tissue? Modern mammals offer several solutions to the problem of cranial protrusions. Giraffes grow ossicones, bony cores permanently covered by furred skin that never breaks through the integument. Rhinoceroses form horns entirely from keratin, with no bony core whatsoever. Bovids, cattle, sheep, goats, antelopes, grow permanent bony cores that become covered with tough, rigid sheaths made of keratin, the same protein forming hair, nails, and hooves. For decades, many reconstructions followed what seemed a conservative middle path, depicting Arsinoitherium horns covered in ordinary skin like giraffe ossicones, or perhaps slightly thickened hide. This interpretation had certain appeal because it avoided assumptions beyond what fossils directly preserved. Without soft tissue evidence, why not default to simple skin?
Museum mounts and illustrations showed horned with surfaces resembling facial skin, perhaps textured but fundamentally unremarkable. This conservative approach faced challenges as researchers examined fossil horn cores more critically. The vascular traces bore striking resemblance to patterns on modern bovid horn cores. Remove the keratin sheath from cattle or goat horns and examine the underlying bone, and you find exactly the same features. Oblique foramina angled into bone surfaces, anastomosing vascular channels running in various directions, overall texture speaking of intimate contact between bone and overlying structure. The similarity extended beyond superficial appearance. The spacing of foramina, depth of grooves, even how vascular patterns change from horn base to tip, all matched the bovid pattern remarkably well.
This observation led several researchers to propose that Arsinoitherium horns more likely bore keratinous sheaths similar to bovids rather than simple skin. The first published description noting the hollow structure appeared in 1903 from an anonymous source, followed by paleontologists Charles William Andrews in 1906, who suggested the horns may have borne keratin coverings in life. For decades, this observation sat in literature without generating much discussion. Later researchers occasionally acknowledged the possibility, but often hedged their bets, presenting both interpretations as equally plausible. The debate intensified in the early 21st century as new generations of paleontologists and paleoartists scrutinized these details with fresh eyes and new techniques. Detailed photography under various lighting conditions highlighted surface features with unprecedented clarity. Some argued forcefully for skin-covered horns, noting that not all mammalian cranial structures necessarily bear keratin. Others championed the keratin sheath hypothesis, marshalling comparative anatomical evidence from living animals. Consider the functional implications. Bone covered by keratin sheaths develops characteristic roughened surfaces where sheaths anchor to cores. Keratin does not simply sit atop bone like a cap. It interdigitates through numerous projections and depressions that lock the materials together. This creates the oblique foramina and complex vascular channels observed on Arsinoitherium horn cores. Bone covered by ordinary skin typically shows much smoother surfaces with less elaborate vascular traces, as skin does not require the firm anchoring that weight-bearing keratin sheaths demand. The keratin sheath, though made of dead tissue like fingernails, forms incredibly tough and resilient structure. It distributes impact forces across underlying bone, preventing localized stress concentrations that could cause fractures. It provides hard, wear-resistant surfaces capable of withstanding repeated impacts. Only with such protection could the delicate, hollow, bony cores have functioned as anything more than visual display structures. The keratin would have supplied durability the thin-walled bone cores alone could never provide. Recent detailed examinations have revealed just how closely Arsinoitherium horn cores match the bovid pattern. The resemblance extends to subtle details unlikely to arise by chance. This has convinced many specialists that the keratin sheath interpretation represents not just possibility, but probability, the most parsimonious explanation for observed anatomy. Yet debate continues. Some researchers point out that Arsinoitherium belonged to a completely different mammalian order than bovids. Could we be certain embrithopods used the same biological strategy? Might alternative explanations exist for vascular patterns that do not require invoking keratin sheaths? These fair questions remind us of inherent limitations when working with incomplete fossil evidence. We cannot perform experiments on Arsinoitherium horns, cannot watch them grow, cannot analyze their chemistry. We can only compare what fossils show to what we observe in living animals and make our best inference about which modern analog provides the closest match. The growth pattern offers another window into structure. Young individuals had much smaller horns than adults, and examining cores from juveniles through fully mature animals traces how these structures developed over lifetimes. The horns grew continuously, adding new bone at their bases and extending length over years. This growth pattern differs fundamentally from deer antlers, which grow and shed annually, but matches well with both giraffe ossicanones and bovid horns, which are permanent fixtures, developing throughout life. Interestingly, the smallest horn pair above the eyes may provide an independent test. These knob-like structures show similar vascular traces to large anterior horns, suggesting whatever covered one pair probably covered the other. Consistency demands applying the same interpretation to all four horns, not just the dramatic anterior pair. If we accept that Arsinoitherium horns bore thick keratin sheaths, then living horns would have appeared even larger and more impressive than bony cores alone suggest. Keratin can add substantial girth and length. In some bovids, keratin sheaths make horns appear nearly twice as thick as underlying bone cores. A living Arsinoitherium might have looked even more imposing than skeletal reconstructions indicate, with truly massive horns dominating the facial profile. The keratin would also have changed texture and potentially color, though without preserved pigments we can only note that modern horned mammals display considerable variation in horn coloration. The texture would have been distinctly horn-like rather than skin-like, with the hard, almost plastic quality characteristic of keratinized structures.
For paleoartists attempting to bring Arsinoitherium back to life, this debate presents genuine dilemma. Should they follow the weight of anatomical evidence pointing toward keratin sheaths, even though this interpretation remains debated? Or take the more conservative route, depicting skin-covered horns, avoiding commitment to hypotheses that might later be overturned? Different artists have made different choices, which is why books and museum displays show both versions. Each approach represents defensible position given current evidence, though the tide of scientific opinion has been shifting toward the keratin sheath interpretation in recent years as new analytical techniques and comparative studies have strengthened the case. The resolution matters beyond aesthetic accuracy. It affects interpretation of structural capability, influencing how we understand what these animals could physically accomplish with their horns. Horn cores covered only in skin, even thickened skin, would remain vulnerable to damage during vigorous contact. The thin, bony cores could fracture if struck with force. But keratin-sheathed horns transform from potentially fragile ornaments into structures capable of withstanding substantial mechanical stress. This structural difference has profound implications for interpreting the behaviors we will explore in the next chapter, where we examine theories about combat, display, and sexual selection. The horn enigma of Arsinoitherium reminds us that even with excellent fossil material, even with complete skeletons and detailed anatomical knowledge, fundamental questions about appearance and function can remain tantalizingly unresolved, subject to ongoing investigation and debate as new evidence emerges and new analytical techniques become available.
Chapter 3: Swamp Dweller or Forest Browser. Imagine standing at the edge of an ancient Egyptian swamp 35 million years ago, watching the shoreline where water meets land. Will Arsinoitherium emerge from the murky depths like a hippopotamus, water streaming from its massive body as it hauls itself onto the bank? Or will you spot it deeper inland, moving through the forest undergrowth like a rhinoceros, browsing on leaves and fruits among the trees? This seemingly simple question about where Arsinoitherium actually spent its time has generated one of the longest-running debates in the study of this extinct giant, a controversy that touches on everything from how we interpret fossilized bone structure to what we can really know about the daily lives of vanished animals. For most of the 20th century, the semi-aquatic interpretation dominated scientific thinking about Arsinoitherium. When researchers looked at the animal's skeleton, particularly the structure of its limbs, they saw an animal that seemed poorly designed for life on land, but beautifully adapted for a life spent largely in water. The evidence appeared compelling. Those sturdy, columnar legs that so closely resembled elephant limbs seemed almost too heavy and inflexible for efficient terrestrial locomotion. More tellingly, the forelimbs showed a peculiar specialization.
The shoulder blade was remarkably wide and flat, much broader than you would expect for an animal of this size, and the muscle attachment points suggested that the primary motion of the front legs was not a forward swing as in walking or running, but rather a powerful backward pull. This anatomical arrangement made perfect sense if you imagined Arsinoitherium wading through shallow water or hauling itself through sticky mud. The animal could have pulled strongly with its front limbs, generating forward propulsion while its broad feet distributed its weight across soft substrates, preventing it from sinking too deeply into the muck. The comparison to modern hippos seemed almost irresistible. Like Arsinoitherium, hippos spend their days largely in water, possess barrel-shaped bodies, have relatively short limbs compared to their bulk, and venture onto land primarily to feed. The parallel extended even to behavior. Hippos are notoriously aggressive when defending their aquatic territories, and those massive hollow horns on Arsinoitherium's skull could have functioned perfectly well in aquatic combat, with males clashing in shallow water much as hippos do today. The geological context of the fossils seemed to support this aquatic interpretation. The Fayum Depression in Egypt, where the most complete Arsinoitherium specimens were discovered, yields fossils from the Jebel Qatrani Formation, a sequence of sedimentary rocks that accumulated in distinctly watery environments. During the late Eocene, this region lay much closer to sea level than it does today. The sediments themselves tell a story of coastal swamps, mangrove-lined tidal flats, sluggish rivers, and lagoons where brackish water mixed marine and freshwater influences. These are not the clean sands of beaches or the sordid gravels of swift mountain streams, but the fine muds and silts that settle from standing or slowly moving water, layer upon layer accumulating in low-energy depositional settings. When paleontologists found Arsinoitherium fossils entombed in such sediments, it seemed natural to conclude that the animal had lived and died in these very environments, spending its days half submerged in the warm, shallow waters of Eocene Africa. Other anatomical details seemed to fit the aquatic lifestyle. The placement of the nostrils relatively high on the skull could have allowed Arsinoitherium to breathe while mostly submerged, with only the top of its head breaking the water surface, another hippo-like trait. The robust skeleton, built more for strength than speed, matched what you might expect in an animal that relied on water for support rather than needing to carry its full weight on land. Even the apparently weak hind limbs made sense in this context. An animal that spent most of its time in water would not need powerful hind legs for running or climbing. The water itself would provide support and enable movement with minimal energy expenditure. When paleontologists found Arsinoitherium fossils entombed in such sediments, it seemed natural to conclude that the animal had lived and died in these very environments, spending its days half submerged in the warm, shallow waters of Eocene Africa.
For decades, this interpretation appeared in scientific papers, museum labels, and popular books about prehistoric life. Artists depicted Arsinoitherium wallowing in swamps, only its head and the upper curve of its back visible above murky water. Some reconstructions showed it feeding on aquatic vegetation, pulling up water plants with that muscular upper lip. The semi-aquatic Arsinoitherium became canonical, the accepted understanding of how this animal had lived. Then, beginning in the late 20th century and accelerating into the 21st, this comfortable consensus began to fracture. Researchers started looking more critically at the evidence and asking whether the anatomical features that supposedly indicated aquatic habits might be explained in other ways. The challenge came from multiple directions, from detailed studies of tooth wear, from chemical analysis of fossilized bone, and from a more nuanced understanding of what those seemingly aquatic adaptations actually meant. The teeth told a story that did not quite fit the aquatic narrative. Arsinoitherium possessed highly specialized dentition, with 44 teeth arranged in continuous rows that allowed for a unique chewing mechanism. The jaw joint could shift position during chewing, maintaining constant pressure along the entire tooth row as the animal processed its food. This sophisticated apparatus was not designed for the kind of tough, fibrous aquatic plants that modern hippos consume. Instead, the tooth shape and wear patterns suggested selective browsing on soft vegetation, the kind of tender leaves and fruits you would find on bushes and low trees in a forest, not growing in a swamp.
When researchers examined Arsinoitherium teeth under magnification, they found wear patterns consistent with a diet of relatively soft plant material. The cusps showed polishing rather than the heavy pitting and scratching that comes from eating gritty, abrasive foods. This dental evidence pointed toward an animal that carefully selected what it ate, choosing the most nutritious and easily digestible parts of plants rather than bulk feeding on whatever aquatic vegetation was available. Modern hippos, by contrast, are far less selective, grazing on grasses and other plants with little discrimination. The comparison that had seemed so compelling at the superficial level started to break down when examined in detail. The chemical composition of Arsinoitherium bones provided another line of evidence. When animals eat and drink, they incorporate isotopes of various elements into their bones and teeth. The ratios of different isotopes vary depending on what the animal consumes and where it obtains its water. Aquatic and semi-aquatic animals show distinctive isotopic signatures that differ from those of terrestrial animals. Researchers, including Clements and colleagues, extracted samples from Arsinoitherium fossils and subjected them to stable isotope analysis, measuring the proportions of carbon-13 to carbon-12 and oxygen-18 to oxygen-16 with exquisite precision. The results surprised many paleontologists. The carbon isotope ratios in Arsinoitherium bones matched those of terrestrial browsers feeding on forest vegetation rather than aquatic plants, which show distinctly different photosynthetic pathways and thus different isotopic signatures. The oxygen isotope values suggested the animal obtained most of its water from the food it ate and from drinking surface water, not from spending extended periods submerged. When compared to the isotope ratios from confirmed aquatic animals found in the same Jebel-Qutrani deposits, Arsinoitherium clearly fell into a different category. The chemical fingerprint preserved in 35-million-year-old bone was telling researchers that this animal had lived primarily on land, not in water. Paleontologists also began to reconsider what those supposedly aquatic anatomical features actually meant. Yes, the forelimbs were structured for powerful backward pulling, but this does not necessarily indicate an aquatic lifestyle. Elephants, which are undeniably terrestrial animals, have similar forelimb structures adapted for pulling down tree branches and manipulating vegetation. The broad shoulder blades and strong pulling muscles could just as easily have served Arsinoitherium as tools for feeding, allowing it to pull branches within reach or tear down vegetation, rather than propelling it through water. When compared to the isotope ratios from confirmed aquatic animals found in the same Jebel-Qutrani deposits, Arsinoitherium clearly fell into a different category. The chemical fingerprint preserved in 35-million-year-old bone was telling researchers that this animal had lived primarily on land, not in water. Paleontologists also began to reconsider what those supposedly aquatic anatomical features actually meant. Yes, the forelimbs were structured for powerful backward pulling, but this does not necessarily indicate an aquatic lifestyle. Elephants, which are undeniably terrestrial animals, have similar forelimb structures adapted for pulling down tree branches and manipulating vegetation. The broad shoulder blades and strong pulling muscles could just as easily have served Arsinoitherium as tools for feeding, allowing it to pull branches within reach or tear down vegetation, rather than propelling it through water. The graviportal limb structure, those columnar legs built like pillars, also required reinterpretation. Graviportal means weight-bearing, and it describes the limb anatomy of extremely large terrestrial animals that need robust skeletal support to carry their mass on land. Elephants are graviportal. So were the enormous sauropod dinosaurs. The limb structure of Arsinoitherium, rather than indicating aquatic habits, might simply reflect the engineering constraints of building a multi-ton animal that could move on land. The fact that the animal could not run swiftly does not mean it lived in water. It might merely mean that an animal of this size did not need to run, because few, if any, predators would dare attack a healthy adult. The broad feet of Arsinoitherium could have distributed weight on land just as effectively as in swamps, preventing this heavy animal from compacting soil excessively as it moved through forests and across floodplains. Elephants, after all, manage perfectly well on firm ground despite their wide feet and relatively flat-footed stance. Even the fossil context became subject to new interpretation. Yes, Arsinoitherium remains were found in sediments deposited in watery environments, but this does not necessarily mean the animal spent its life in those environments. Many terrestrial animals live near water, drinking from rivers and lakes, feeding on vegetation that grows most abundantly in well-watered areas, and occasionally crossing waterways as they move through their territories. When they die, their bodies often end up in water, carried there by floods or by their final approach to drink. The taphonomy, the process of how organisms become fossils, can create misleading associations between animals and depositional environments. River systems and coastal swamps are excellent places for fossilization precisely because they bury carcasses quickly in fine sediments, but the animals preserved there may have spent most of their lives on adjacent dry land. More Arsinoitherium fossils from different locations complicated the picture further. Specimens found in Ethiopia came from sediments that represented somewhat drier, more seasonal environments than the coastal swamps of Fayum. If Arsinoitherium required permanent water bodies and a fully aquatic lifestyle, it should not appear in these drier settings. Yet there it was, adapted to a range of environmental conditions that suggested considerable ecological flexibility. The animal could survive in various habitats, from the wettest coastal areas to more open, seasonally dry woodlands.
So where does this leave us? After more than a century of study, we find ourselves with two competing interpretations, each supported by some evidence, each challenged by other data. The traditional semi-aquatic view cannot explain the dental evidence, the isotope data, or the occurrence in drier environments. The newer terrestrial interpretation struggles with some of the anatomical specializations, particularly those forelimbs that truly do seem better suited for poling than for walking, and must explain why an animal so obviously adapted for living near water, at minimum, would not spend at least some time actually in that water. The most sophisticated current thinking suggests that Arsinoitherium probably occupied a middle ground, an animal that was fundamentally terrestrial in its basic ecology, but maintained a close association with water. Think perhaps of a modern forest elephant, which spends most of its time on land browsing in dense vegetation, but regularly visits water holes, wallows in mud, and is perfectly capable of swimming when necessary. Or consider the tapir, another large forest-dwelling herbivore that lives on land but frequently bathes and feeds near water. Arsinoitherium may have been similar, a forest browser that concentrated its activities near rivers, swamps, and coastal areas where vegetation grew most abundantly, that perhaps used water for cooling and wallowing as elephants do today, but that did not spend the majority of its time submerged like a hippopotamus.
This interpretation reconciles most of the evidence. The animal could browse selectively on forest vegetation, explaining the teeth and isotope data, while still benefiting from those powerful forelimbs when crossing muddy ground or moving through dense swamp vegetation near the forest edge. It could have lived in a variety of habitats from wet to moderately dry, as the fossil distribution suggests, while still showing anatomical adaptations that made wetland environments particularly suitable. The skull could remain high and dry most of the time while the animal fed on land, but those strong legs could still support wading through shallow water when necessary. The daily life of Arsinoitherium, if we accept this intermediate interpretation, would have been spent moving through forests, selecting the most nutritious leaves, fruits, and shoots it could find. During the heat of the day, it might have rested in shade near water, perhaps occasionally wading in to cool off or escape biting insects. In drier seasons, it would have concentrated near permanent water sources where vegetation remained green. In wetter times, it could range more widely through the landscape. The massive body size meant that adult Arsinoitherium had little to fear from predators. The contemporary meat-eating mammals called creodonts that shared its environment would have found a multi-ton giant an intimidating prospect. The lifestyle question matters because it affects how we understand virtually every other aspect of Arsinoitherium biology. If it was aquatic, then those horns might have been used primarily in water, with males battling in rivers and swamps. If it was terrestrial, combat would have occurred on land, perhaps in forest clearings or at territorial boundaries. Social behavior might differ between an animal tied to water sources and one that could range freely across the landscape. Even the reasons for its eventual extinction depend partly on whether we are looking at an animal specialized for wetlands or one adapted to a broader range of conditions. The ongoing debate about Arsinoitherium's lifestyle teaches us something important about the nature of paleontological inference. We can examine fossils in extraordinary detail, measure isotope ratios to parts per thousand, compare anatomical structures with living animals, and analyze depositional environments with sophisticated geological techniques. Yet after all this effort, fundamental questions about how an extinct animal actually lived can remain genuinely uncertain. Different lines of evidence point in different directions, and the extinct animal cannot be observed directly to settle the dispute. This is not a failure of science, but rather an honest acknowledgement of the limits of what we can know about vanished worlds. The mystery of Arsinoitherium's lifestyle, terrestrial browser, or semi-aquatic inhabitant, or something in between, may never be completely resolved. But the process of investigating the question, marshaling evidence, testing hypotheses, and refining our interpretations brings us closer to understanding this remarkable creature and the ancient African ecosystems it inhabited.
Chapter 4. Combat, Display, and Sexual Selection. Stand before that mounted skeleton in a museum and ask yourself the most obvious question that Arsinoitherium's anatomy provokes. Why? Why invest so much biological capital in growing a pair of enormous horns that could reach half a meter in length or more? Why construct these elaborate hollow towers of bone, vascularized and maintained throughout life? Why burden the skull with structures that, even in their lightweight configuration, added substantial mass to the front of the head? The answer, as with so many dramatic anatomical features in the animal kingdom, almost certainly lies in the intertwined realms of competition, reproduction, and social signaling. When Charles William Andrews first examined complete Arsinoitherium skulls in the early 1900s, he noticed something curious about the specimens arriving from Egypt. They did not all look quite the same. Some skulls bore horns that seemed more robust, more heavily constructed, with thicker walls and more pronounced curvature. Other skulls, similar in overall size, and apparently from fully adult animals, had horns that appeared somewhat more gracile, slightly smaller in cross-section, occasionally less dramatically curved. For a time, some researchers interpreted these differences as evidence of multiple species living side by side in the same environment. Perhaps there was a larger, more heavily armed Arsinoitherium species, and a smaller, more delicately built one, both inhabiting the coastal forests and swamps of Eocene Egypt.
But as more specimens accumulated and researchers had the opportunity to study larger sample sizes, an alternative explanation emerged. What if these were not different species at all, but simply males and females of the same species? Sexual dimorphism, the condition where males and females of a species differ significantly in size or appearance, is extremely common in mammals, particularly in features related to competition and display. In modern animals, we see it everywhere. Male deer grow elaborate antlers while females remain antlerless. Male lions develop magnificent manes that females lack. Male elephant seals grow to sizes that dwarf their mates. When competition for reproductive success is intense, evolution tends to exaggerate the differences between the sexes, particularly by elaborating the features males use to compete with each other or attract female attention. The pattern observed in Arsinoitherium skull collections fits this sexual dimorphism interpretation remarkably well. If we accept that the more robust skulls with larger, more heavily constructed horns belong to males and the more gracile specimens to females, suddenly the variation makes perfect sense. Both sexes possessed horns, all four of them, but males invested more heavily in horn growth, producing structures optimized for the intense male-male competition that characterized their breeding system. Females had horns too, suggesting these structures served some function beyond pure male competition, perhaps in defense against predators or in establishing dominance hierarchies among females. But they did not need the extreme development seen in males.
This interpretation transforms how we understand Arsinoitherium social behavior. Instead of solitary animals wandering through the forest independently, we can begin to envision a species with complex social dynamics, where males competed vigorously for access to females, where dominance mattered, and where those impressive horns served as both weapons and advertisements of quality. The question then becomes, exactly how did males use these horns in competition, and what did females look for when choosing mates? The anatomy of the skull and neck provides crucial clues. When researchers examine the rear of an Arsinoitherium skull, they find an unusual configuration of bones and muscle attachment sites that speaks volumes about how this animal moved its head. The occipital region, where the skull connects to the neck vertebrae, shows evidence of truly extraordinary muscular development. The bones here are thick and robust, with pronounced ridges and crests where massive neck muscles attached. Andrews measured these features precisely and noted that the occipital condyles, the rounded projections that articulate with the first neck vertebrae, were unusually large and positioned in a way that would have permitted powerful vertical movements of the head. The occipital region, where the skull connects to the neck vertebrae, shows evidence of truly extraordinary muscular development. The bones here are thick and robust, with pronounced ridges and crests where massive neck muscles attached. Andrews measured these features precisely and noted that the occipital condyles, the rounded projections that articulate with the first neck vertebrae, were unusually large and positioned in a way that would have permitted powerful vertical movements of the head. Most tellingly, the structure suggests that Arsinoitherium could have performed forceful upward swings of the head with tremendous power. Picture the motion. An animal with its head lowered, those forward-projecting horns aimed at an opponent, suddenly throws its head upward in a violent arc. The leverage generated by thick neck muscles pulling on the back of the skull would have driven those horn tips upward with considerable force. This is not the motion of an animal using its horns like battering rams, smashing head-on into opponents. Instead, it suggests a wrestling style of combat, where males would engage horns, lock their weapons together, and then strain against each other in tests of strength, each trying to twist the other's head, throw the opponent off balance, or drive him backward. Modern animals provide instructive parallels. Male oryx, those elegant African antelopes with their long, straight horns, engage in elaborate sparring matches where they lock horns and push against each other, testing strength without usually causing serious injury. Male bighorn sheep clash their massive curled horns together with spectacular crashes that echo across mountain valleys, the impacts absorbed by specially thickened skull bones and neck muscles. Male giraffes swing their heavy heads like clubs, using their short horn-like ossicones to deliver powerful blows to opponents' bodies and necks. Each species has evolved a fighting style that matches its anatomical tools. For Arsinoitherium, the evidence points toward a wrestling approach. Two males would approach each other, perhaps after a period of visual display and vocal intimidation. They would lower their heads, bringing those forward-swept horns into position, and then engage, horn against horn. The hollow structure of the horns, which might seem fragile, would have been reinforced by those keratinous sheaths we discussed in an earlier chapter, providing the durability needed for vigorous combat. Once engaged, the males would push, pull, and twist, each trying to gain leverage over his opponent. The powerful neck muscles would come into play, allowing sudden upward thrusts or sideways wrenches that could unbalance a rival. The spacing between the horn tips, that 40-centimeter gap, takes on new significance in this context. It was wide enough that when two males engaged, their horns could interlock in various configurations, allowing for the complex wrestling moves that would determine the victor. A male with longer horns might have an advantage in reach, able to make contact before his opponent could close distance. A male with thicker, stronger horns might withstand twisting forces better. The exact shape and size of a male's horns would have determined his effectiveness in these contests.
But combat, while certainly part of the story, probably does not tell the whole tale. Those horns were visible long before any fight began, and in the world of sexual selection, appearances matter enormously. A male Arsinoitherium walking through the forest bore his horns like banners, advertising his quality to any female or rival who saw him. The size, symmetry, and condition of those horns conveyed information about the male's genetic quality, health, and age. Growing and maintaining enormous horns required resources, and only in an environment with abundant food could males afford such extravagance. A male needed to consume enough nutrients not just to build the bony cores, but also to produce the keratin sheaths that covered them, to maintain the vascular tissues that kept the horns healthy, and to carry the weight throughout his daily life. Only a vigorous, healthy male with access to high-quality food resources could afford truly impressive horns. A male struggling to find enough to eat, or one burdened by parasites or disease, would have to allocate his limited resources to basic survival rather than extravagant ornament. His horns might grow, but they would be smaller, less perfectly formed, perhaps asymmetrical or poorly maintained. Females observing males in the population would have immediately perceived these differences. A male with magnificent horns announced his quality from a distance, signaling that he possessed good genes, occupied a territory with abundant resources, and was strong enough to thrive despite the burden of his ornaments. A male struggling to find enough to eat, or one burdened by parasites or disease, would have to allocate his limited resources to basic survival rather than extravagant ornament. His horns might grow, but they would be smaller, less perfectly formed, perhaps asymmetrical or poorly maintained. Females observing males in the population would have immediately perceived these differences. A male with magnificent horns announced his quality from a distance, signaling that he possessed good genes, occupied a territory with abundant resources, and was strong enough to thrive despite the burden of his ornaments.
This is the essence of sexual selection through mate choice, an evolutionary force that Darwin himself recognized and wrote about extensively. When females preferentially mate with males who display certain traits, those traits become exaggerated over generations even if they serve no survival function, and might even impose costs on their bearers. The peacock's tail is the classic example, absurdly elaborate and costly to grow and carry, yet maintained by evolution because peahens prefer males with the most impressive displays. Arsinoitherium horns likely evolved under similar selective pressure, with females choosing mates based partly on horn quality, thereby ensuring that each generation of males invested even more heavily in these structures. The hollow internal structure of the horns introduces another fascinating possibility. Some researchers have suggested that these air-filled chambers might have functioned as resonating cavities, amplifying vocalizations and allowing males to produce louder, more impressive calls. Recent studies of Arsinoitherium inner ear anatomy have revealed adaptations for hearing low-frequency sounds, similar to modern elephants, suggesting these animals may have communicated using infrasound that could travel long distances through dense forest. If Arsinoitherium males used their hollow horns to augment such vocalizations, they could have produced calls of extraordinary volume and distinctive quality. The exact frequency and character of the sound would have depended on the size and shape of the horn cavities, meaning that each male had a unique acoustic signature determined by his horn morphology. Females could have assessed males not just by looking at their horns, but by listening to the sounds they produced, with larger horns generating deeper, more resonant calls that carried farther through the forest. We can imagine the breeding season, whenever that occurred in the Arsinoitherium annual cycle.
Males would have ranged through the forest, bellowing challenges to rivals and advertisements to females, their calls amplified by those remarkable hollow horns. When two males encountered each other, visual display would have been the first level of interaction. They would have sized each other up, comparing horn size and body condition, each assessing whether his opponent was worth fighting or whether discretion suggested retreat. Many contests would have been settled at this stage, with the smaller or less impressive male withdrawing without physical contact. When males were too closely matched for simple display to settle the matter, escalation to actual combat would have occurred. The wrestling matches we described would have ensued, violent shoving matches that tested strength, endurance, and fighting skill. Injuries were probably relatively rare, as is the case in most mammalian species where males possess dangerous weapons. Natural selection favors ritualized combat that determines winners without routinely killing or crippling the participants. Dead or seriously injured males contribute nothing to the gene pool, so evolution tends to establish rules of engagement that limit damage. The fights would have ended when one male acknowledged defeat, perhaps by breaking contact and retreating, or when one male succeeded in forcing his opponent to the ground or driving him from the contested area. The winner gained access to receptive females, earning the reproductive payoff that made all the investment in horns worthwhile. The loser retreated to try his luck elsewhere, his horns intact to fight again another day. Female Arsinoitherium, watching these contests or assessing males independently, face their own evolutionary calculus. By choosing to mate with males who won fights or who displayed the largest, most symmetrical horns, females were selecting genes that would give their offspring, particularly their sons, advantages in future competitions. The sons of successful males would inherit the genetic potential for growing impressive horns, making them more likely to succeed in competition when they reached maturity. This created a positive feedback loop, with successful males producing successful sons, and females who chose such males producing more grandchildren than females who mated randomly. Female Arsinoitherium, watching these contests or assessing males independently, faced their own evolutionary calculus. By choosing to mate with males who won fights or who displayed the largest, most symmetrical horns, females were selecting genes that would give their offspring, particularly their sons, advantages in future competitions. The sons of successful males would inherit the genetic potential for growing impressive horns, making them more likely to succeed in competition when they reached maturity. This created a positive feedback loop, with successful males producing successful sons, and females who chose such males producing more grandchildren than females who mated randomly. The presence of horns in females, even if smaller than those of males, suggests that these structures served functions beyond pure sexual display. Female Arsinoitherium likely used their horns in defense, particularly in protecting calves from predators. The contemporary carnivores of Eocene Africa, large creodonts with powerful jaws and formidable hunting abilities, would have found young Arsinoitherium tempting prey, and even healthy adults might have faced threats from pack-hunting predators. A mother defending her calf could have used her horns to threaten or even attack a predator, and the forward-projecting configuration would have been effective for driving away a threat. Females may also have established dominance hierarchies among themselves, with horn size and fighting ability determining access to the best feeding areas or the safest resting spots. In modern elephants, closely related to Arsinoitherium through their shared Afrotherian heritage, females live in matriarchal social groups, where older, larger females dominate younger ones. If Arsinoitherium females maintained similar social structures, the horns would have played a role in establishing and maintaining their social positions within the group. The small posterior horns above the eyes, so easily overlooked in favor of their dramatic anterior neighbors, may have played a distinct role in communication. Positioned prominently on the front of the face, these knobs would have been highly visible during frontal displays. Some researchers have speculated that they might have been brightly colored or that they could have supported skin flaps or other soft tissue ornaments that enhanced their visual impact. Without fossilized soft tissue, we cannot confirm such details, but the strategic positioning of these small horns suggests they mattered in visual communication, even if they contributed nothing to combat effectiveness. The evolution of Arsinoitherium's horn complex represents a textbook case of runaway sexual selection, where female choice and male-male competition drive the elaboration of traits far beyond what would be favored by natural selection alone. From a pure survival perspective, smaller horns or no horns at all might have been optimal. The animal could have avoided the energetic costs of growing and maintaining these structures, could have moved more easily through dense vegetation, and would have had better forward vision unobstructed by its own weaponry. But sexual selection does not optimize for survival alone. It optimizes for reproductive success. And in the competitive world of Arsinoitherium breeding, males without impressive horns simply did not get to pass on their genes. This same evolutionary dynamic has shaped countless other species, from the antlers of deer to the tusks of elephants to the elaborate plumage of birds of paradise, producing extravagant traits that serve no purpose other than attracting mates and deterring rivals. Understanding the horns as signals, as tools of combat and display shaped by sexual selection, brings Arsinoitherium to life in ways that dry anatomical description never could. We see not just a skeleton, but a social animal, one that competed, displayed, chose mates, and invested heavily in the evolutionary games that ultimately determine which genes persist and which disappear. The forest of Eocene Egypt would have echoed with the bellowing calls of males, would have witnessed the violent wrestling matches that established dominance, and would have seen females making the careful assessments that shaped the evolution of these magnificent creatures. The horns tell all of these stories, if only we know how to read them.
Chapter 5. When the Rains Stopped Falling. For millions of years, Arsinoitherium thrived across a warm, green Africa that would be unrecognizable to anyone standing on that continent today. During the late Eocene epoch, roughly 36 to 30 million years ago, the Sahara Desert did not exist. Where today we see endless sand dunes and barren rock, there once stretched continuous forests, networks of rivers, and vast coastal swamps. The climate was humid and warm, with abundant rainfall supporting lush vegetation year-round. From the northern coast along the Tethys Sea down through what is now Egypt, Libya, and across to the Ethiopian highlands and south into regions that would become Angola, this was Arsinoitherium country, an empire of plenty where these massive herbivores ruled as some of Africa's largest land animals. For millions of years, Arsinoitherium thrived in the warm, forested Africa of the late Eocene, those lush coastal swamps and river systems where it browsed on soft vegetation and used its impressive horns in competition for mates. The stable, humid environment shaped every aspect of its biology. The elaborate horns, costly to grow and maintain, could evolve precisely because resources were so abundant that males could afford to invest heavily in sexual display and combat. The specialized dentition, designed for selective browsing rather than bulk feeding, worked perfectly where high-quality food items were always available. Even the relatively slow, deliberate locomotion posed no disadvantage when there was nowhere the animals needed to rush to, and when predators, though present, could be deterred by sheer size and those formidable horns. But planetary climate does not remain static forever, and Earth was about to undergo one of the most significant environmental transitions in mammalian history. This transformation, known to geologists as the Eocene-Oligocene transition, would fundamentally reshape climates worldwide, triggering extinctions across multiple continents and opening ecological opportunities for entirely new groups of organisms. The late Eocene marked the beginning of a long-term cooling trend that would change ocean circulation, atmospheric patterns, and terrestrial ecosystems on a global scale. The causes were complex and interrelated. The positions of continents continued to shift as plate tectonics rearranged Earth's surface.
Antarctica, which had been gradually separating from South America and Australia for millions of years, finally achieved complete isolation. Ocean currents that had previously flowed in different patterns reorganized themselves into the circumpolar Antarctic current, isolating the southern continent in a ring of cold water that would eventually lead to the formation of the vast ice sheets that still cover Antarctica today. The growth of Antarctica ice had global consequences. Ice reflects sunlight back into space rather than absorbing it, a phenomenon called the albedo effect. As ice accumulated on Antarctica, the planet's overall temperature began to decline. Ocean temperatures dropped. The amount of water locked up in ice meant that sea levels fell. Atmospheric circulation patterns shifted as temperature gradients between the poles and the equator changed. The greenhouse gas concentrations in the atmosphere also declined during this period, particularly carbon dioxide levels, removing some of the atmospheric blanket that had kept the planet warm. The growth of Antarctic ice had global consequences. Ice reflects sunlight back into space rather than absorbing it, a phenomenon called the albedo effect. As ice accumulated on Antarctica, the planet's overall temperature began to decline. Ocean temperatures dropped. The amount of water locked up in ice meant that sea levels fell. Atmospheric circulation patterns shifted as temperature gradients between the poles and the equator changed. The greenhouse gas concentrations in the atmosphere also declined during this period, particularly carbon dioxide levels, removing some of the atmospheric blanket that had kept the planet warm. For organisms living through this transition, the changes did not happen overnight. Climate shifts on geological timescales, and what we describe as rapid change might unfold over hundreds of thousands or even millions of years. Arsinoitherium did not wake up one morning to find its world transformed. Instead, generation after generation experienced subtle shifts. Perhaps the dry season, if there was one, lasted a bit longer than it had in their grandparents' time. Perhaps the wettest months delivered slightly less rain. Perhaps the geographic range of certain preferred food plants shifted gradually, retreating from areas that had become marginally less suitable. Initially, these changes might have been imperceptible or easily accommodated. Arsinoitherium was not locked into a single narrowly defined habitat type. Fossils from different localities show that the genus could tolerate some environmental variation. This ecological flexibility suggests that early in the cooling trend, Arsinoitherium populations could adjust their ranges, shifting gradually to track their preferred habitat conditions as those conditions moved across the landscape. The fossil record provides hints of this adjustment. Arsinoitherium remains from the Oligocene epoch, which began 34 million years ago as the Eocene ended, show up in regions that were somewhat drier and more seasonal than the lush Eocene coastal swamps. The animals persisted, clearly capable of surviving in environments that were not quite as wet and warm as their ancestors had enjoyed. The discovery of Arsinoitherium giganteum in Ethiopia from deposits dated to around 27 million years ago demonstrates that at least some populations survived for several million years into the Oligocene. But survival in increasingly marginal habitat is not the same as thriving. As the climate continued its relentless shift toward cooler and drier conditions, the fundamental character of African ecosystems began to change. The transformation was most dramatic in northern Africa, the region where Arsinoitherium had been most abundant. The coastal forests and river systems that had sustained rich mammalian communities started to contract. Rivers that had flowed year-round became seasonal, drying to scattered pools during increasingly long dry seasons. Forests fragmented into isolated patches, separated by more open terrain. The mangrove swamps along the coast retreated or disappeared altogether as sea levels dropped and coastal geography changed. Most significantly for Arsinoitherium and many other forest-adapted animals, grasslands began to spread. Grasses, which had been relatively minor components of Eocene plant communities, found the new, drier, more seasonal climate ideal for their growth strategy. They spread rapidly across regions where forests could no longer maintain themselves. This was not yet the dramatic expansion of savanna that would characterize the later Miocene epoch, but the first stirrings of what would become one of Earth's great biomes were already underway. For Arsinoitherium, this shift spelled catastrophe. The animal's entire biology was optimized for a forest environment with abundant soft vegetation. Those specialized teeth, so perfect for processing tender leaves and fruits, were poorly suited to the tough, silica-rich grasses that were taking over. The animal could not simply switch diets the way some more generalized herbivores might. Evolution had refined Arsinoitherium into an elegant machine for exploiting a particular ecological niche, and that very refinement became a fatal limitation when the niche itself began to disappear. We can trace the decline in the fossil record, though the picture is frustratingly incomplete, as fossil records always are. Arsinoitherium remains, common in late Eocene deposits across northern Africa and Arabia, become progressively rarer in Oligocene sediments. The geographic range appears to contract, with the latest occurrences concentrated in areas that retained forested conditions longer than surrounding regions. The Ethiopian highlands, with their orographic rainfall patterns that could sustain forests even as lowland areas dried out, harbored some of the last populations. The Aragalette beds in Kenya, dated to between 27.5 and 24 million years ago, have yielded what are now recognized as the final chapter in Arsinoitherium's story, the very youngest fossils of this lineage known to science. These Kenyan deposits represent the last occurrence of embrithopods in the entire fossil record. After that, silence. By the Middle Oligocene, around 25 to 24 million years ago, Arsinoitherium vanishes from the fossil record entirely. The last of the embrithopods, those strange African relatives of elephants that had evolved their rhinoceros-like body plan in splendid isolation on the island continent, had finally succumbed to environmental changes they could not overcome. Why could they not adapt? Why did Arsinoitherium disappear while other lineages survived the Eocene-Oligocene transition and went on to flourish in the new environmental conditions? The answers lie in the constraints imposed by their own evolutionary history. Adaptation is not infinitely flexible. An organism cannot simply decide to develop new characteristics in response to changed conditions. Evolution works with the raw material provided by existing anatomy, physiology, and behavior, modifying them gradually through variation and selection. Some starting points offer more evolutionary potential than others. Consider the comparison with the proboscideans, those elephant relatives that were evolving alongside Arsinoitherium in Eocene Africa. Early members of the elephant lineage, animals like Moeritherium and Phiomia, also lived in wet, forested environments and faced the same climatic changes that challenged Arsinoitherium. But the proboscidean body plan contained within it the seeds of future success. Their flexible trunks, initially perhaps used for aquatic feeding, could be repurposed for gathering food in a variety of habitats. Their continuously replacing molars, where new teeth grew in at the back of the jaw and moved forward as older teeth wore out, allowed them to process increasingly tough vegetation without losing their ability to feed. Their large body size and complex social behavior gave them the flexibility to range widely across landscapes, finding scattered resources. Arsinoitherium had none of these advantages. The specialized chewing mechanism that had been such an asset in forest environments offered no path toward processing grass. The animal could not simply evolve new teeth. Such a fundamental change would require a complete reorganization of skull architecture, something that happens over millions of years, if it happens at all, and Arsinoitherium did not have millions of years. The locomotor system was not designed for the long-distance movements that would become necessary as resources became scattered across increasingly arid landscapes. The enormous horns, shaped by sexual selection in an environment where males could afford costly ornaments, represented a massive energy investment that made less and less sense as food became harder to find. Population size likely played a role as well. As suitable habitat contracted, Arsinoitherium populations would have become smaller and more fragmented, isolated in scattered forest refugia separated by expanding grasslands. Small populations face genetic risks, losing variation through drift and becoming more vulnerable to inbreeding depression. They are more susceptible to local extinction from random events like drought, disease outbreak, or simply bad luck in the sex ratio of offspring. And crucially, small populations produce fewer variants, the raw material on which natural selection acts, reducing the probability that somewhere in the species, some individual will possess the genetic variants needed to survive in changed conditions. The extinction of Arsinoitherium was not an isolated event, but part of a broader reorganization of mammalian communities worldwide during the Eocene-Oligocene transition. Many lineages disappeared during this period, particularly those adapted to warm forested conditions. The ancient creodonts, those predatory mammals that had dominated carnivorous niches earlier in the Cenozoic, declined dramatically, eventually replaced by the ancestors of modern carnivorans, the cats, dogs, bears, and their relatives. Numerous primitive ungulate groups vanished. The transformation created opportunities for new lineages to radiate and diversify. But for those like Arsinoitherium that were too specialized for the old world, there was no place in the new.
The final populations of Arsinoitherium, clinging to shrinking forest patches in the Ethiopian highlands or isolated river valleys in East Africa, faced a slow but inexorable slide toward oblivion. Each generation would have found fewer suitable feeding areas, more pressure from competitors better adapted to the changing conditions, longer distances to travel between remaining patches of appropriate habitat. Young animals might have struggled to find mates as population density declined. Perhaps females produced fewer offspring, or offspring survival rates dropped as environmental stress mounted. However the end came, it was probably not a sudden catastrophe, but a gradual fading, populations winking out one by one until no more remained. What might have saved them? In an alternate history where climate had stabilized rather than continuing its cooling trend, where forests had persisted across northern Africa, Arsinoitherium might have continued indefinitely, might even still walk the earth today as an African megafaunal icon alongside elephants and rhinos. If the genus had possessed greater dietary flexibility, if it could have learned to process the grasses that were spreading across its former range, it might have made the transition to the new grassland ecosystems. If it had evolved a different locomotor strategy allowing greater mobility, it might have been able to shift its range to track retreating forests. But evolution deals in what is, not what might have been, and the specific trajectory that Arsinoitherium had followed left it vulnerable when the world changed. The empty niche left by Arsinoitherium's disappearance would eventually be filled by other large herbivores. True rhinoceroses, members of the Perissodactyla that had evolved on northern continents, would eventually disperse into Africa and occupy the role of large horn-bearing browsers, a remarkable case of ecological replacement where unrelated lineages converge on similar roles in different times and places. But for several million years after the last Arsinoitherium died, Africa's ecosystems lacked the particular combination of size, diet, and habit that this genus had embodied. Today, Arsinoitherium exists only as bones and teeth entombed in ancient sediments, museum specimens that speak eloquently of vanished worlds, and as a reminder of evolution's fundamental truth: specialization brings both success and vulnerability. For millions of years, being exquisitely adapted to warm, wet, forested Africa made Arsinoitherium one of the most successful mammals on the continent. When Africa changed, that same exquisite adaptation became a death sentence. The horns that had intimidated rivals and impressed mates could not reach up to pull down clouds and make rainfall again. The specialized teeth that had efficiently processed forest vegetation could not somehow remake themselves to handle grass. The powerful limbs that had carried multi-ton bodies through swamps and forests could not outrun climate change. The story of Arsinoitherium's extinction is ultimately a story about the limits of adaptation and the contingency of survival.
Species do not persist because they deserve to or because they are somehow superior in an abstract sense. They persist because they happen to possess characteristics suited to their current environment, and they vanish when that environment changes faster than they can evolve. This ancient extinction carries lessons that resonate powerfully in our own time. In a world currently experiencing rapid climate change driven by human activity, where habitats are fragmenting and species adapted to specific conditions are finding those conditions disappearing, the fate of Arsinoitherium offers a sobering parallel. The magnificent horned giants of Eocene Africa remind us that even successful, well-adapted animals can prove surprisingly fragile when the ground shifts beneath their feet, when the rains fail, when the forests they depend on simply cease to exist. They thrived for millions of years, and then they were gone, leaving only scattered bones as testimony that they ever walked this earth at all.
Arsinoitherium's story reveals fundamental truths about evolution and extinction. This elephant relative, disguised as a rhinoceros through convergent evolution, thrived for millions of years in the forests of ancient Africa. Its spectacular horns, shaped by sexual selection and possibly covered in keratin, made it one of the continent's most impressive creatures. Yet, the very specializations that brought success, teeth designed for soft vegetation, a body built for forest life, became fatal limitations when climate shifted toward cooler, drier conditions. The parallels to our current moment are inescapable. As human-driven climate change accelerates, countless species face the same dilemma that doomed Arsinoitherium. Can they adapt fast enough to survive a rapidly changing world? These ancient giants remind us that even millions of years of evolutionary success offer no immunity to environmental upheaval. Evolution deals in what is, not what might have been, and specialization, while powerful, always carries hidden costs. Thank you for listening to this exploration of one of evolution's most remarkable deceptions and cautionary tales.